And he's Dan Flippin Science looking at cell differentiation, methylation, and epigenetics.
If we ever come close, if we ever discover how to hold on and keep our faith in each other, the difference between us will keep us together.
So science understands when we look at cellular differentiation associated with tissue growth and development is controlled by gene expression. Recognize that changes in DNA methylation and histone modification can alter gene expression. Epigenetic change can lead to phenotypic differences between identical siblings. Phenotypic differences between clones may cause human diseases. And explain how epigenetic modifications in genes that control cell division, such as changes in DNA methylation, can lead to cancer. So in organisms, there's a hierarchy of organization. So cells need to coordinate to produce a whole organism. So we have cells of one or more similar types grouped together to form tissues, different tissues grouped together to form organs, and different organs together to form systems. And then lots of systems together make your organism. So here's a visual representation of that. So we have different cell types. So we have muscle cells, nerve cells, epithelial cells, connective cells. They combine with very similar cells or cells of the same type to produce tissues. Several different tissues combine together to produce an organ. In this case, we're looking at a kidney. So there's muscles in kidneys, there's nerves in kidneys, there's lining cells, epithelial cells in kidneys, and also connective tissue. And then organs can join together to make up a system. So in this case, we've got the urinary system, and then lots of different systems together to make combine together to make the organism. So there's a question from this. If all the cells in an organism have the same genetic information, how do we get different types of cells? Since the cell division we're looking at, so mitosis, is asexual, all the daughter cells produced are identical. I've got a video here that goes through showing how zygotes form blastocysts. So a zygote is what happens after egg cell is fertilized. Blastocyst is after many cell reproductions have occurred. So I'm going to go through this a bit quicker. So here we've got our initial cell. It divides into 2, and then those 2 divide into 4, and those 4 divide into 8, those 8 divide into 16, and so on and so on and so on. So as time goes on, you end up with more and more cells, and those cells will eventually start to differentiate, which means they're not exactly the same. They're not phenotypically the same, even though they're genetically the same. So you end up at the end with a big ball of cells that we call the blastocyst. So if we look at an example of this in the zygote, you have 3 layers of tissue. You have the ectoderm, which is the outside, mesoderm, which is the middle layer, then you have the endoderm on the inside. So very early on in the life of a human, when we're looking at very small numbers of cells, we get differentiation of these layers. So the cells produced here are slightly different. And then they begin to fold and so on, and eventually they end up with some of the structures that are very important. So we have the notochord, so that is basically what turns into the nerves inside the spinal column. You have this neural tube, which turns into the spinal column, and you have other layers as well. So when I talk about differentiation, what I mean is the changing or specializing of cells into particular roles. So the question is, if all the cells are produced from mitosis and therefore they're genetically identical to each other, how do you get the cells turning into different types of cells and doing different roles? And the answer is that only a few genes are turned on in any particular cell. So you can have some genes turned on and then therefore other genes are turned off, and that means that different structures and functions can be produced from cells with the same genetic information. So here's some examples of some differentiated cells. So all of these cells, if they're coming from the same person, will have the same genetic information, but different genes being turned on or off will lead to different structures. So for example, we have the axon in a nerve cell. We have the round biconcave disc of the blood cell. We have white blood cells being very different in structure from red blood cells, even though, again, they're produced from the same genetic information. Then we have all the epithelial cells, like the cheek cells we looked at under your microscope. So all of these have the same genes, it's just some genes are turned on, some genes are turned off, and that leads to different structures and functions. So just summarizing that, turning on or off genes is responsible for the differentiation of cells into different structures and functions. So let's talk about epigenetics. Epi means above, so we're kind of going above the genetics that you normally have. It can be caused by environmental factors, but it's not always. What this means is that during your life, you can accumulate changes in your DNA that can then be passed on to the offspring. So they're heritable changes in your gene expression based on how you live your life and exposure to certain things in the environment sometimes, but also there's also non-environmental causes. So it doesn't involve changing the underlying DNA sequence. So your DNA sequence stays the same, but it changes how the DNA is read. So we're changing the phenotype without changing the genotype. And this can affect how cells read genes and the expression of genes. So some examples of this, surrogate mothers and genetically unrelated offspring can have, so in a surrogate mother you have Unrelated sperm and egg fertilized into a zygote inserted into a mother who becomes a surrogate. The experience of the surrogate mother during pregnancy can impact the epigenetics of the offspring that's being produced. This has happened in horses, for example, so we've got some evidence for this from horses. So we're going to talk about methylation first. So chemistry students should know what a methyl group is, so it's a carbon with 3 hydrogens attached to it. All cells have the same genetic information in there, so all the DNA is the same if we ignore things like mutations that pop up. But the DNA itself in different types of cells can have different structures and functions. And one of these causes of this is a process called methylation. This is where a methyl group, so a carbon and 3 hydrogens, is attached to cytosine nucleotides just before a gene starts. So here is the nitrogenous base of cytosine, so we would have attachment to the sugar coming off of the carbon over here. By adding the methyl group, it makes it harder for the gene to be transcribed. And the reason for this is that transcription factors, enzymes and bits and pieces that come together to lead to transcription, so copying the gene into mRNA. When you have methylation, those factors and enzymes, they can't read the DNA, they can't attach around the gene, so the gene doesn't get transcribed into mRNA. And that means because you're not producing the mRNA, you're not producing the protein or the RNA molecule that would be produced from the reading of the transcribed mRNA, so you don't get the effect of the gene. So let's have a look at this in some pictures. So here we have a gene that is activated, and if we had a look, it's unmethylated. So we have unmethylated bits here before we get to the gene, and that means that we're going to get lots of expression here. If we have a look at this gene here that's being repressed, we can see we've got methylated regions of the bit just before the gene, which is called a promoter, and this means those transcription factors won't be able to attach and the gene won't be read. And here's another example over here, so just showing you what it looks like. So we have these little islands, which are your promoter regions. If it's unmethylated, like we have over here, then we're going to have nothing blocking the transcription factors leading to transcription, so the gene's going to be expressed. Whereas over here we have this whole methylated region over here, and here, that means that the gene won't be able to be read. It also goes back the other way, so DNA can be demethylated, and that means that you're going to get more expression of the gene. So if we look at this direction, so the cytosine here, so this is where we attach the sugar down here, we remove the methyl group, and then that means that we're removing the block to the transcription, so the gene can be expressed. So if we have a look here, so here's our methylated gene, so we've got our methylated island before we start our gene. If we demethylate it, then the gene can be expressed. So we're going to get our expression of the gene and its characteristics. Histone modification is also another way of doing this epigenetic regulation. So we can modify the histones, which are those proteins associated with DNA, to either reduce or encourage the expression of genes. Again, we have methylation. So if we have methylation, we get the histones sticking together, and that means it's very hard for the transcription factors to attach to the DNA and the expression to occur. So that's down here. So we have methylation, so we've got methyl groups that are being attached to histones. That means they stick together more. It means the DNA doesn't unwind as much, so we're not going to get transcription factors attaching to it. Acetylation is the opposite. So we had methylation and demethylation. Here we have acetylation in histones. So with acetylation, we get looser packing. So we can see we have acetyl groups being attached. So that means the DNA is much more easily exposed to the transcription factors and enzymes. That means you're going to get more expression of the genes. So here's another picture showing that. So here we have methyl groups that are being attached to the histones. You can see the histones are really close together, and here's our gene that we're trying to transcribe, and it can't be accessed by the transcription factors. Down here we have acetylized histones, so we can see that the histones are much further apart, they're spread out, and the gene over here that we're trying to express is accessible.
So where can this happen? Choices of things like stress during your life, diet, lifestyle, taking drugs or smoking, the amount of sport and things like that you can do, all of these can have an effect on the epigenetics, and then they can be passed on to your offspring. Now these epigenetic changes, they occur pretty much as soon as the zygote can be produced. This means that there can be differences in gene expression between identical twins, for example. So there can still be differences in phenotype in identical twins, even though their genotype is identical due to this methylation. And also in cloning too. So in cloning, again, you have the same genetic information between your two clones, but due to these epigenetic changes over the course of the clone's life, they can lead to changes in phenotypic expressions. Epigenetics can also lead to cancer. So what cancer is, you have, if we're looking at, this is skin, for example, we're looking at skin cancer here. You have your regular cells, and then if you have one cell that switches over to not being able to regulate its reproduction, it starts reproducing over and over and over, you get tumors being produced, and that's what a cancer is. So cell division is usually regulated. So your cells don't just keep on reproducing and reproducing, they only reproduce when they're really required to. So there are genes that are involved in doing this regulation. If methylation occurs and these suppressor genes are turned off, then there won't be transcription occurring, and that means that suppressor proteins that would normally reduce the amount of reproduction occurring won't be produced. So there's less suppression. If there's less suppression, that means there's less control of cell division, and therefore you get more production of cells, and that can eventually lead to tumors. So some disorders where this is related to, we can have Fragile X syndrome, Prader-Willi syndrome, and Angelman syndromes. They can all be produced from this methylation. So the methylation switches off genes that would produce suppressor proteins that would stop cells from constantly reproducing, so the cells start to constantly reproduce.
So let's do our check. So we talked about cellular differentiation and how we get different types of cells based on different genes being expressed within different cells. We talked about methylation and histone modification, how that can alter the expression of genes. We talked about how epigenetic changes can lead to phenotypic differences between identical siblings, but also between clones, and it may cause human disease. And we talked about how epigenetics can possibly lead to cancer by affecting the methylation of genes that produce factors that might inhibit cancerous reproduction of cells. That's it for this understanding. See ya.